Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. In the last couple of classes, we have been looking at various questions in the form of tutorials, and let us continue for this, maybe one more class also based on this particular thing. Okay, towards the end in the previous class, we were looking at some examples. Let's say look at this particular question. In a metalloprotein, you were told that the metal ion is bound to histidine 51 and glutamic 55, but you are asked to prove this. Okay, you are given a wet biochemical, biological, microbiological based labs. What is your strategy and how will you prove this? Yeah, so the fact that is given the position, so that means you need to sort of take this position into a different kind of amino acid. So, how do you change one amino acid by other, and that is what is referred as the site targeted mutagenesis. So, therefore, by using the site targeted mutagenesis, you can replace one at a time, the histidine 51 at one time, glutamic 55 at another time. You can also do both of them as well, multiple mutations too. And from that you can definitely prove that the reactivity is present, reactivity is not present, all these things. For this you require a lot of PCR work and the nucleic, the plasmid based work. That is why we talked about biochemical and biological kind of a work. Then once you change this, you have to express. So, you express in microbugs. So, therefore, microbiology. Then you synthesize, you get the product which is in the form of the enzyme, and that enzyme activity you can study. So, that is how one can approach for such a kind of a problems. Okay, so the next question, the tutorial, because these tutorials are supposed to be making you to think as how to respond to the questions. So, you are given a metalloprotein and an absorption spectrometer. I would say you are given different metalloproteins and you are given a absorption spectrometer and a wet chemistry lab. So, what all can you obtain by using all these and how? So, when you say wet chemistry lab, you should be able to do chemistry modifications. For example, you are given a metalloprotein, you can take out the metal by using some chelate agents. Then you get a apoprotein, you can check the apoprotein absorption spectrum, then you start adding the metal ion. So, as a function of the metal ion concentration, as a function of the pH, all of these you can study. So, from that you can establish the metal ion, its coordination aspects such as geometry, other things, and many of these kind of things can be identified. So, therefore, the same question will come in another form later on. So, we will look into that as well. Okay. So, these two what I talked now, actually I have already talked in the previous class towards the end. Okay, let us look at the next one. You are given a metalloprotein containing copper plus, and you are asked to find how many histidines are bound, okay, to that metal ion center in that protein. So, you are given only one spectroscopy instrument. So, you decide which one you wanted to use and a wet chemistry lab, which means you can play with it. So, which spectroscopy instrument will you choose for this activity and how you are going to establish the number of histidines bound to copper plus center in that given protein. So, you are allowed two things, any one spectroscopy technique, and you are allowed to do some chemical modifications too, because chemistry lab is given. So, suppose you want to go, suppose you think that your answer is absorption spectroscopy. See, the absorption spectroscopy is not going to tell the number of histidines at all. You may say, argue that the shifts of these very broad bands, suppose you have a few histidines or more histidines, the positions could shift to the very similar to that. So, therefore, absorption spectroscopy is not a right method, rather EPR. How will I say EPR? It is copper plus. Everyone knows copper plus is D10, and D10 is a non-magnetic one. But try to see in the third line a wet chemistry lab. So, what I do? I will add some agent which converts the copper plus to copper 2 plus. So, what will I use? I can use an oxidizing agent. So, I will oxidize to copper 2 plus. Once it is oxidized, I have a copper 2 plus, which is a D9 system, and now this D9 system I can tackle through the EPR spectroscopy. And if I go for a high resolution EPR spectroscopy, I can even establish the number of histidines being present in this one, and that is how it is. Okay. So, let us keep looking at more and more questions of involved type. What simple technique that you would recommend if one were to establish the molecular weight of a protein? Can the same technique be used to find whether the protein is pure or any impurities are present? How? First of all, to find molecular weight, the best technique is mass spectrometry. And for protein, what is the best mass spectrometry? Is the MALDI, matrix assisted laser desorption ionization spectroscopy. So, therefore, obviously MALDI, and the MALDI sensitivity is so high, it can identify even the concentrations of 10 power minus 9, minus 10 in that range too. So, if you have a impurity which is at low concentrations than your protein, still you can find out. So, therefore, MALDI is absolutely a good thing. I am not just telling answer, I always, for every question I am giving the reasoning. So, you should also think first the reasoning of this. Okay, let us move on to the next query, query in the tutorial. A carboxylic derivative of 18 crown 6 ether shows increasing transport rates for calcium 2 plus as a function of pH, increasing pH. So, it is a carboxylic derivative, it is 18 crown 6 and it increases as a function of pH for calcium. Whereas, this is exactly reverse for potassium plus. What that means? As you increase the concentration of the potassium plus instead of calcium 2 plus, so it goes down. Okay. So, to explain this observation. Now, what we know is what happens when the pH is added or pH is increased, pH is increased for a carboxylic group. What will happen? At very highly acidic pH, carboxylic group will be COOH, and as you keep increasing the pH, slowly some portion of carboxylate will come. At neutral, almost most of the thing could be carboxylate, and in a basic medium also it will be completely carboxylic. So, that self is a half of the answer, or in other words main clue has come. So, that means the calcium is binding very well when you increase the pH. When you increase the pH, you have more and more of carboxylate, and we have studied in the chapter that carboxylic groups have a very specific affinity towards calcium, and in fact nature has modified even glutamic acid to one additional carboxylic group being present. The same thing at low pH, the potassium plus will have a better transport, because at low pH it is completely COOH, and COOH is neutral, and that binds better to potassium plus, and that is the reason absolutely. So, always try to analyze the question and try to understand what we know first, then what we wanted to discuss and debate. Let us take the next question. So, assume that a metal can either occupy a tetrahedral or occupy a octahedral site with similar ligating groups in a metalloprotein. Let us assume that. Suggest a simple spectral method that could differentiate these two possibilities and explain as how it would differentiate between these two geometries. I have already explained to you absorption spectroscopy, where tetrahedral versus octahedral. Tetrahedral versus octahedral. Tetrahedral gives a completely allowed transitions, therefore you have a very high absorption coefficient, whereas octahedral it is both spin as well as Laporte forbidden, therefore you will have a very little absorptivity. So, therefore, one can easily find out by using absorption spectroscopy whether the metal ion has gone into a cage of the octahedral or whether into a core of a tetrahedral. So, tetrahedral versus octahedral can be easily found out by absorption spectroscopy. Okay, now let us look at more and more questions based on each metal enzyme kind of thing. So, let us look at this particular slide. The first these are based on the vanadium. Okay, so let me read the first question on the top. In this, while vanadium shows -1 to +5 oxidation states in its compound, These are restricted to +5, +4, +3 in biological systems and none other, not even +2. Discuss and throw your views on it. So, we know if when you open the chemistry compounds, chemistry aspects of vanadium, what you will find? Vanadium, you will find all kinds of oxidation states -1 to +5. You know the carbonyls etc. will show 0 and -1, minus, whereas the other compounds can show plus, and oxides can show +5 and some extent +4, halides etc. etc. So, therefore, in chemical compounds you have all through -1 to +5, but whereas in biological system it is only +5, +4, +3. Why? So, that is the question. So, that means there must be some problem if it goes to +2. If it goes to +2, what could be the problem? It will immediately reduce this environment or surrounding and get self oxidized to +3 or +4 or even +5. So, therefore, the lower oxidation state are strongly reducing. Therefore, the system, biological system does not accept such kind of an extreme kind of things, and hence I think it is reasonable to say that this is the reason why the +3, +4, +5 only are used in the biological systems, not so much so +2 and lower oxidation states. Okay, let us look at the next question. The vanadium 51 NMR is well suited for identifying its primary coordination sphere. What oxidation state is suited for this? So, obviously, any NMR when you look for a non-paramagnetic or preferably the one which is diamagnetic. So, which of the vanadium is diamagnetic? Vanadium 5. Why? Vanadium 5 is D0. Okay. What and how the primary coordination sphere is identified? Shine light on it. See, if you go back and look into the story that I have explained on vanadium in biology, one plot you will see x axis is the vanadium chemical shifts having 0 on one side minus, other side plus, and the y axis you have a sigma chi by n. Sigma chi is the total the electronegativity of all the atoms by n is the number of ligating centers or the coordination number, and that you have seen pretty much. So, you go to more and more negative, more and more electronegative atoms, the value will be more and more negative, and the more and more positive will have the positive. And now I will say, I will not give you all the answer. I have activated you to look into that particular plot, and you can get with the answer very nicely and easily. So, now if you know the vanadium 51 NMR, you can draw simply a line from the y axis onto the straight line and try to get what kind of a groups are there on this. Yes, I hope you can do that, and it is not very difficult. You should be able to do that. Let us move on to the next one. Though the entry of 5 vanadium in biological system is +5 oxidation state, that is the one. So, what are the kind of species +5 oxidation states? Vanadate and other vanadates too, depending upon the concentration and pH. Guess what kind of forms? How is that one identifies +4 and +3 species state, okay, in the cellular and biological fluid? So, discuss this phenomena and shine its reasonings. See, actually, though the entry in the biological system is +5, when it gets inside the cells, they get into 4, they even get into 3, because there are cellular reducing agents. The cellular reducing agents will reduce this one. So, these can be identified by EPR when it comes to the vanadium 4, which is D1, can be absorption come EPR also +3, all these kinds of things. When you have +5, you can use from the NMR. So, variety of these techniques can be used and identified. So, it is basically the cellular reducing agents which will convert +5 to +4 or +3, and they are stabilized by cellular molecules, they are complex by cellular molecules after being reduced. So, therefore, their existence in the cell is not only 5, can be +4, can be +3. Okay, let us get to the next one. What is the negative role or inhibitory role played by vanadium in biological systems? Does this in terms of discuss this in terms of the what, how and why. Okay. So, negative role, I am sure you understand, in the movies negative role is by the villain. So, it is the same thing here too. So, what kind of an inhibitory role? In case a negative role in biological systems is nothing but the inhibitory role. So, you know, for this the answer is that you need to compare vanadium as vanadate and the phosphorus as the phosphate. And why we have to compare? Because it is the phosphorylation which is most important to make the enzymes to be active, which I have told several times during variety of enzyme reactions when we are discussing. Therefore, in the case of the sodium potassium ATPase, as you know, in the second cycle where the potassium binds and the enzyme undergoes a dephosphorylation, at that stage if the vanadate is found is present, the vanadate will bind to the enzyme and make a irreversible contact with the enzyme. Therefore, vanadate will be acting like a inhibition in the second part of the cycle. And this also I have given in the course in one of the slides. You can always see and try to understand that too. Looks like there are some more questions in my slides. Let us go into that. So, in one protein, a vanadilyl VO2 plus species is bound by two nitrogen centers. Okay. So, you have a VO and VO is bound by the two nitrogen centers. Where are the nitrogen centers? Primarily histidines, and it is also written in the question as histidines. What spectral technique would you use to confirm that the vanadyl center is in this case is bound by two histidines? No, absolutely. So, VO2 plus, what is the oxidation state of vanadium? It is 4 plus. What is the d electron system? D1. So, D1 is a best system for EPR and no other complication. And if you go for a high resolution EPR, the nitrogens which are bound, which is having I is equal to 1, will further split, and from the number of lines you can make that. The same kind of a question has come earlier in different form. So, the answer is the same. Let us come to the next question. In biological systems, in one case vanadium 5 species is bound by 4 oxygen ligands, VO4 binding core. In another, the vanadium 5 is bound by 2 oxygens and 2 nitrogen ligands, VO2 N2 binding core. So, what is the technique that you would use to differentiate this? Just a while ago I talked to you vanadium 51 NMR. I talked to you it is dependent on the summation of the electronegativities. When you have all 4 nitrogens, the summation of electronegativities is greater as compared to the VO2 N2, 2 oxygens and 2 nitrogens, the summation of chi is different. Therefore, from the straight line you can easily find out, and their NMR positions are absolutely different. Therefore, you can differentiate. Okay, go to the next question. There are 2 redox reactions are given. VO2 plus 2H plus electron giving rise to V3 plus H2O. The potential is plus 0.3. Okay, let us get to the next question where we have a redox potentials VO2 plus 2H plus electron V3 plus H2O, which has a plus 0.3, and the other equation given is VO2 plus 2H plus electron VO2 plus 2H2O, which is the 1 electron volts. So, what is the first? Identify the couple in A, also in B, compare both and tell which one of these redox couples is more favored and why. So, for this what you need to look at? You need to look at first of all what is the couple. So, on the left side VO2 plus, vanadium is 4 plus, right side is V3 plus, it is a couple V4 plus versus V3 plus. In the next one VO2 plus, okay. So, that is what 4, then plus is there 5, vanadium 5, and then on this side VO2 plus, vanadium is 4. So, 5 to 4. So, 5 to 4 is plus 1, 4 to 3 is plus 0.3, that means 5 to 4 is more favored as compared to the 4 to 3, but both are favored because both are positive. And this is, you have to take delta G is minus NFE, since the potentials are positive it will be still minus. So, more negative value, less negative value, 5 to 4 is more negative value, 4 to 3 is less negative value of delta G, therefore, their corresponding the feasibilities, etc. Okay, let us get into another arena of the enzymes, which is manganese based. Look at the one of the questions first. In a photosystem related to plants, what is the gross reaction that occurs at photosystem 1 and that occurs at the photosystem 2? So, it is already given in my slides. At the photosystem 1, you need to do reduction and bring the carbohydrate. In the photosystem 2, you need to do the oxidation of the water to O2. So you can easily do that. So, what is the chemical nature of these reactions? The first one is reduction, the second one is oxidation. So, in photosystem 1 always you have a reduction, and photosystem 2 you always have a oxidation. Go to the next question. Given the fact that one single manganese ion can undergo a change from +2 to +7 oxidation, we know very well, even from our high school, the teacher tells that the manganese has a large number of oxidation states, and in the school level we study this +2 to +7. You have other things are also possible. Why does the oxygen evolving complex require 4 manganese ions? First of all, what is the oxygen evolving complex? Is the photosystem 2, which is a manganese complex. So, what is this reaction? This reaction is basically oxidation, that is 2H2O giving rise to O2 plus 4H plus 4 electrons. So, totally how many electrons? 4 electrons. So, 4 electrons change if you have one manganese, if it is in 2, it can go to 3, it can go to 4, it can go to 5, it can go to 6, or if it is in 7, it can come to 6, 5, 4, 3. All of these are possible with one manganese, but it does not happen. It happens by 4 manganese ions, because in this particular enzyme, each reaction is by one pulse of light, and each pulse of light will activate one proton, one electron. And therefore, these are all transferred to the each metal ion that you have. Therefore, the whole thing is taken care by all the 4 manganese ions, not by the one single. So, therefore, one requires this. So, not just one. Of course, where the actual reaction occurs, it is one of the manganese and a calcium center. So, between this calcium center and one of the manganese, the entire conversion of water is 2 OH2s, 2 waters. The OO has to come closer, the OO has to make a single bond, the OO has to make a double bond. The hydrogens which are attached to O should go away, etc., etc. Then all 4 protons should be out, then electrons should be in to get the bond, and both the things are there. Okay, what is the total reaction that occurs with the manganese superoxide dismutase? For any superoxide dismutase, it is basically the O2 minus. So, it is not 1, it will give 2 of this, which will give 1 O2 and 1 H2O2, 1 O2 and 1 H2O2. So, in some enzymes, first there will be a reduction, then in the second step oxidation. In some enzymes, first there is a oxidation, next there is a reduction. This is how it happens. So, it can be in the manganese, it can be in the iron, it can be in the copper, etc., etc. In all the cases, this is what the basic story is, and entire thing very clearly given in my slides. I refer you to look back into the slides in these things. Every one of these. So, you can listen to my lecture followed by you can use the one. There are some more queries of the manganese in biology. While the dismutation of superoxide is affected by one manganese center, the disproportionation of the hydrogen peroxide needed 2 manganese center. So, what is dismutation of superoxide? It is called superoxide dismutase. What is the disproportionation of the hydrogen peroxide? It is called catalase. So, you have the superoxide dismutase catalase. So, how many electrons in superoxide dismutase? 1 electron. How many electrons in peroxide? 2 electrons. Therefore, you have one metal center in case of superoxide dismutase, which can take care of the 1 electron, and 2 electrons have to be taken care. Therefore, you have 2 manganese centers, and that is what. And that is what will happen. So, both the electrons simultaneously will be transferring, things can happen. Okay. All right. In the next question, looks like in the previous question itself I extended the answer, but we can look at again. At photosystem 2, the 2 water molecules are converted to O2 by the 4th manganese, that is the manganese that is located outside the Cubian structure. If you look at my slide, you will find and calcium 2 plus. These two, these are the ones. If there were, if that were be the case, why should there be a tetramanganese cluster? I have already answered to you. Give you reason and then explain this in the context of the functioning of this enzyme at each pulse of the light. I have already explained to you that the manganese undergoes each time one oxidation state, and that is transferred to the neighbor manganese systems, etc., in order to have a total catalytic. And second thing is, for each pulse of the light there is a proton and there is an electron being generated and utilized, and these are being very well taken care with when you have the tetranuclear cluster. But actual reaction occurs between the fourth manganese and the calcium, and that is what we are looking at. So, that is where the reasons that we are given. Okay, let us see in the next question. In photosystem 2, the O2 water molecules are converted to O2. Give your own philosophy to make this conversion happen, not using the slides given in the class. Design a hypothetical scheme showing the principles and approach that you would wish to adopt. So, to form a O2 from H2O and H2O, first of all, what you have to do? You have to bring the two O's of the H2O close by. Then what you have to do? You have to disconnect the hydrogens. When you disconnect one hydrogen from this, one hydrogen from the other water, then you can get the one bond. Okay? So, one proton out, one electron, one proton out, one electron, the two electrons is one bond. In the next stage, one more hydrogen is removed as a proton, and for other water also it is removed, therefore, one more bond, second bond. So, therefore, you will find, you will make two bonds. So, this means I need to make an enzyme system or enzymatic model system where the two metal ions are holding two oxygen waters and they are in close proximity. And if I can put some kind of a conformational change to this, of course, I am almost mimicking the enzyme, and that is how it is. Okay? So, I hope you understand in that. Okay, let us start with some more tutorial problems. Let us move into other enzymes too. So, the next one is iron in biology. Iron case, huge amount of iron story I have already talked to you. Let us start looking at this. Though there is about 1% of CO present in cells, the hemoglobin and myoglobin bind selectively to O2 and not to CO. How and why? Let us first look at that part of it. 1% of CO always is present in our cells because the dead cells are decomposed, and that is where the reason where you get 1% of CO. Now, you have a CO binding, you have a O2 binding. At 1% of this is the concentration of the CO is not very high, and the enzyme has a mechanism to differentiate the O2 binding versus CO, which I told in the class. The CO binds straight, O2 binds bend. The bend one is stabilized by a hydrogen bond with a histidine, a distal histidine, and which is not happening by CO. CO is repelled by a carboxylate group. So, in CO case it is repelled, in O2 case it is attracted, therefore, the system goes towards the O2, and therefore, O2 is recognized over CO at those concentrations. However, when the concentration of CO is very large, then there is an equilibrium is shifted and the CO binds. If the CO binds once, it will continue, because CO binding is very strong and is as strong as 30 times to that of the O2. Therefore, the iron CO is more favored, and therefore, iron O2 is not favored. This is what exactly happens when I talked about this story. I also mentioned, must have given the story, that in most of the fire accidents, the death is not because of the fire burning, skin burnings or body burning, rather than it is because of the choking by the CO, because there is a lot of smoke, because you have a smoke which is overtakes this. Okay, so let us look at the next question. You have learned that the globin protein, hemoglobin, myoglobin, prevents the formation of mu oxodimer. What is this mu oxodimer? How is it formed? How does the globin protein prevents its formation, and what should be the end result if not prevented? Mu oxodimer is two iron centers are coming closer and bridged by the oxo, and that is what exactly. So, you have an iron with one O2, another iron on O2. When both these two have come together, it will become initially a iron peroxo iron, then iron oxo iron. Therefore, the iron oxo iron will be the mu oxodimer. And once that is formed, the iron centers are also oxidized to form that one. So, if they are oxidized to form that one, then what do you have? You have no way it will reverse. Therefore, mu oxodimer formation is dangerous. And in case of enzyme, the two such units cannot come closer because the protein covers these two groups and they cannot come closer with a distance of because the two irons have to come within about 3 angstroms or so, they can never come. Therefore, mu oxodimer is not formed and it is being prevented. Okay, assuming that the O2 stretching frequencies of the free and bound to hemoglobin, hemerythrin and hemocyanin are 1400, 1150, 850, 750 respectively. Okay. So, comment on the nature of the O2 binding to those proteins and substantiate your answer accordingly. Okay. So, assume that the stretching frequencies are this one 1400 quite high, little low, much low, very much low in these things. Okay. So, when your O2 is absolutely intact, you will get 1400. When some little disturbance or perturbance is there, it will become 1150. When it is particularly reduced to single bond, then it will become the 850 hydroperoxy type. When it is completely peroxo, then it becomes 750, and that is what happens. So, in the hemoglobin very little perturbation, and the hemerythrin hydroperoxy kind of species, and in hemocyanin peroxy species, which is bridge between the two copper centers. The ion present in the N-terminal and C-terminal lobes of transferrin has difference in their binding strengths. Explain how would you identify the experimentally such a stage. Yes, if you go for release of these N-terminal as well as the C-terminal, you would be able to release one of them at a little higher pH, the other one at a little lower pH. The one which is at lower pH is more stronger, that is happens at the C-terminal. The higher pH will happen at the N-terminal, which is somewhat weaker. And this can be done by electrophoresis, many kind of thing, many methods. This is possible to do all this. Okay, so we have many more questions, and then I will continue as we keep moving on across in the next class or so. Thank you very much.